Hello, everyone. My name is Sandra Flores, and I'm the Field Application Scientist Manager in Europe here at Unchained Labs. I will be your moderator, and thank you for joining us today. We will have a Q&A section at the end of the presentation. To ask questions, all you have to do is click the Q&A button in the Zoom navigation bar at the top or the bottom of your screen, and simply type your question. When submitting a question, please avoid the anonymous button so we can reach out via email if we are unable to get to your question. We will get to as many of them as we can. And now I would like to introduce Nellis Dennis, Product Manager at Unchained Labs. Today, Nellis will talk us through how Lunatic delivers simple, one-step, reagent-free and standard-free RNA quantification, even on samples with DNA or protein contamination. And now I'll hand it over to Nellis. Hello, Nellis.
Hey, Sandra. Thank you for that introduction. Oh, my camera is just giving some technical issue. Okay. I hope everyone can still see me and I'm sharing my screen correct. Okay, thank you everyone for joining now. And Sandra, thank you for that introduction. Let's go and continue on talking about bust out fast, accurate RNA quant, even on messy samples. Although I can live with some chaos, there are limits to how much mess I can accept before it gets in the way. And there are a whole different set of limits that my girlfriend will accept. I think of RNA samples in a very similar way. A few applications can accept some mess in your samples and impurities, but many more of them have very strict limits on sample purity. Samples that are too messy will impact your results, and that's why it's so important to know when to clean things up, and that's where our Lunatic system can help. Lunatic is a full spectrum Vis spectrometer, which requires only 2 microliter of sample and no dilutions or standard curves. Lunatic can measure up to 96 samples at a time in just 5 minutes if you're using our single microcuvette Lunatic plate with limited dynamic range. If you're using our plate, high Lunatic plate with 2 microcuvettes, it will allow reading higher concentrated samples, and that will do 96 samples in about 10 minutes. Our plates have a standard SBS footprint, so they're fully compatible with any liquid handling robots. Lunatic is the only system out there that can measure RNA at high throughput and high concentration. It uses microfluidic circuits with 2 fixed path lengths to cover a wide dynamic range of 0.03 to 275 OD, which is our high Lunatic plate. Once the sample is loaded, it will be pulled into the reservoir area here by capillary action, and when the sample hits a hydrophobic point, it will stop. At this step, the sample can sit for up to 2 hours before reading with no evaporation. And after the plate is loaded into the instrument, the empty microcuvettes will be read first. That will remove any absorbance contribution from the plastic. And then a pump will apply a small vacuum, which will move the sample into the microcuvettes for the actual measurement. Now, fixed cuvettes make for accurate and fast concentrations since we can always be sure of the exact path length that you need to use in Bayes' law's calculations. It also allows us to correct for stray light and other background. Fixed path lengths mean you know exactly what you're measuring. Now, how can we prove that our fixed path lengths are exact? Well, by using 2 path length absorbance standards for microvolume specs, certified by the National Institute of Standards and Technology, or NIST. We're looking at uracil and tryptophan NIST standards, and they are measured for absorbance. And the expected value is then compared to the measured value, of course. If they're close within certain limits, then our fixed path length can be used for accurate and reproducible measurements. And here we're looking at the path lengths measured on 8 different measurements, 8 measured absorbances in 5 different instruments of both uracil and tryptophan standards. And they were all within 1.65% of the nominal path length as certified. And the mean, which is the X here, and the median, which is the line in the middle, were within 1.25%. This means that Lunatic generates data you can really trust. Here we can see exactly what sample loading looks like with the help of some fluorescine and a black light. In 2 wells, you can see that we already loaded 2 microliter of sample, And as you now can see, to load another sample, you simply add in that 2 microliter into that input well, and it will automatically go into the plate. And now we can see exactly how that looks like when it's loaded versus how it's measured. On the left, you can see loaded samples as we just looked at. And on the right, you can see the samples after they've been measured with Lunatic. The 2 diamond shapes from our high Lunatic plate are the cuvettes for 1 sample. What this also shows is that after pumping, all the sample that you measured remains in your consumable. So there's no contact between the sample and the actual pumping system. So there's no contamination or cross-contamination possible. Now, in recent conversations with a customer, they were working with high concentrated therapeutic plasmids and high concentrated RNA. And they were getting very variable results on their own method, on their classic method, because they had to do a lot of sample dilutions. Now, Lunatic for them was a great solution because they could read samples without any dilution. The data shown here demonstrates how Lunatic handles high concentrated samples without diluting, with the same great accuracy and reproducibility. The highest concentration of DNA in this dilution series on the left was 12,800 nanograms per microliter, and the highest RNA concentration on the right was 6,800 nanograms per microliter. And here's a look at linearity, accuracy, and precision. This shows an RNA dilution series of 8 replicates from 1.5 nanograms per microliter up until 875 nanograms per microliter. The close agreement between the target concentration and the actual measured concentration shows off Lunatic's spot-on accuracy within 2% of the expected value, with an R-squared of 0.9999. On the right, we just zoomed in a bit to show how nice it all aligns at the lower concentrations as well. Since DNA and RNA samples can be very messy, I want to show you on the coming slides how Lunatic's unmixed applications let you see annoying impurities that other, maybe more classic UV systems would mistake as DNA or RNA. We're looking here at results from a sample of pure calf liver RNA, so your perfect, pure, and simple RNA. This is a typical result of a clean sample, and we can see the expected peak maximum at 260 nanometers, classic quality ratios from 260 to 280 are close to the expected value of 2 for pure RNA. And I also want to note out the accuracy and reproducibility. We're seeing nice low standard deviations and CVs, so coefficients of variation for octuplicate reads here. Okay, now we're looking at result of an RNA sample that actually has been contaminated by protein. In both graphs, the blue line is the total measured spectrum. And protein contamination could be picked up by checking your 260-280 ratio, but then you still don't know your actual RNA amount. You just know a total amount like it's shown on the left, and that you have a low 260-280 ratio, for example, but you don't know your actual RNA amount. So on the right, we're analyzing the same sample with our unmixed algorithms to deconvolute the total signal into the signal from RNA and the protein and some other impurities. By using unmixed, we know exactly what's going on in our samples, can spot exactly what the problem is, and you can take the right action. This sample would have to be repurified before using it in a downstream application if protein contamination is an issue for you. So don't miss contaminants that can screw up your sequencing. Check your preps on Lunatic and know what's in your sample. Now, if you would have DNA contamination in your RNA, that's a lot harder to see with classic metrics like A260 and A280 because DNA and RNA spectra are so close to each other. So here we're looking again at a result of an RNA sample where we've simulated DNA contamination by spiking in some DNA. Again, in both graphs, the blue line is the total measured spectrum. A classic UV instrument would report this all as pure RNA, which is shown on the left, and give you a result of 487 nanograms per microliter. But we know, since we spiked that in, there's actually DNA contamination. And so when we take a look with our unmixed application, the software returns this as well. showing you actually only have 284 nanograms per microliter RNA. So about 40% less than what you thought. So know exactly what you have with Lunatic. Now, on the previous slide, we looked at one concentration where some DNA was spiked in. Now, to test our capabilities a bit further, we started with an RNA stock of about 500 nanograms per microliter RNA, pure, and we spiked in calf thymus DNA in certain ratios, all to keep a total amount of nucleic acids around 500 nanograms per microliter, but with different ratios of RNA and DNA. So for example, the pure one, 95% RNA and only 5% DNA, 90% RNA and 10% DNA, up until 60% RNA and 40% DNA. Lunatic's measured RNA concentrations drop completely as expected, showing you that our unmix works to detect your DNA contamination, whether you have a ton of DNA or just a little bit. And as a follow-up experiment, because now we just looked at different levels of DNA contamination, but all at about 500 nanograms per microliter nucleic acid as content. So we picked out one of those, the 70% and 70% RNA and 30% DNA sample, and we diluted it in different steps to make sure that our unmixed application can also detect the RNA and DNA at various concentrations. And as you can see in this data, the RNA amount was always nicely detected, and the established RNA content was within 2% of the target 70% RNA content overall in all of these samples. Okay, so we just looked at Vis and how classic systems would either give you a total amount, but not anything about impurities or anything like that. Like a sample could be flagged by a messy sample could be flagged by a 260 to 80 ratio that is low, but you still wouldn't know how much you would have. Now, another option that you would use to quant is a fluorescent dye. So let's take a look at downsides for doing nucleic acid quant with a dye. Fluorescent protocols are multi-step. They require dilutions and they take time, use costly reagents, and can introduce pipetting errors, which will negatively impact your results. This experiment was done on DNA samples, but of course the same concept applies for using ribogreen and RNA. Costly fluorescent dyes are only linear for a short range. At low concentration, Lunatic and picogreen here agree nicely, while at higher concentrations, costly fluorescent dyes will saturate. So ditch the time-sucking dye-based prep, wasteful standard curves, and the risk of being outside your linear range. Know right away if your samples are good to go with Lunatic. And if we compare the cost of those two methods, so your fluorescent dye protocol and Lunatic, in a theoretical workflow for about 50,000 samples, Lunatic will offer significantly lower consumable and labor costs. Consumable cost savings on those 50,000 samples was about $65,000 a year, and the labor savings was about $78,000 for the overall amount of samples. Now, the biggest part of that savings was that Lunatic doesn't use standard curves or any reagents to quantify your nucleic acids. That will save both on your consumables itself and on time in general, since you don't need to dilute your sample or any DNA standards. And don't lose sleep over instrument to instrument variability or lab to lab variation. Lunatic loves repeating itself, so standardizing your methods across all labs is very easy. With boatloads of sample at multiple locations, the last thing you need is to struggle with validating the same workflow over and over again. Here you can see a box and whisker plot with 3 different concentrations of DNA. Either they were run in octuplicate on the same system, or they were run in octuplicate on 8 different Lunatic systems for a total amount of 64 data points per concentration. On one instrument, we've already discussed the amazing results earlier on RNA. Here we can see that with 8 completely different systems, there's also nothing to worry about. Having systems in different labs in the same building or even at different sites globally will always provide the same high quality results. And in addition to everything we just discussed, Lunatic also meets the needs of labs working in larger scale. For your high throughput needs, our plates are perfectly compatible with liquid handling robots, and so they can nicely pipette everything for you, and everything can go fully automated, from plate loading to actually putting the plate in the system and retrieving your results into a LIMS or anything like that. And we also have IQOQ procedures available, and for labs doing manufacturing, quality assurance, quality control, Lunatic has performance verification as well. That's a series of tests that meet the United States Pharmacopoeia and the European Pharmacopoeia guidelines for Vis spectrometers. The tests and all calculations that come with it are automatically handled by the software. And if you would require it, we have a software add-on tool that can assist you in getting 21 CFR Part 11 compliance that will provide full audit trails, electronic signature capabilities, and anything like that. So to conclude, a messy pile of laundry makes it hard to know exactly what you have to wear. You always need to see things more clearly to sort everything out and know what you have. That's just how Lunatic helps you see what's going on to know when it's time to clean up your messy RNA. And thank you for your attention. And let's see if Sandra has any questions for me now. You're still muted, Sandra.
Okay, sorry about that. Thank you, Nellis, for taking us through how to use the Lunatic to accelerate nucleic acid quantification with one-step measurements, accurately quantifying RNA across a broad range of concentrations, even if they aren't pure, besides saving time and money over fluorescence-based methods. We have some very good questions here that have already been submitted. Remember, you can still ask a question by entering it in the Q&A section of your Zoom navigation bar. So let's start with the questions that I can see here. Nellis, what other kind of samples can I quantify with Lunatic?
Well, basically anything that absorbs. This was very RNA focused, of course, but it remains a UV-Vis system, so you can measure DNA, you can measure protein, yeah, anything that absorbs, rather. Okay, great, fantastic, thank you. And you mentioned that, but it's good as a refresher. What is Lunatic's dynamic range? So if you're using our high Lunatic plate, it will be in optical density from 0.03 to 275 OD, and that translates in 1.2 nanograms per microliter RNA up until 11,000 nanograms per microliter RNA. Okay, thank you very much. What I have next here, what impurities can the mix feature detect? So now in this presentation, everything shown was with the RNA any source application the moment we're using Unmix. So next to DNA and protein, which it will do simultaneously as well, you can detect phenol, azide, and thiocyanate salts as well. All right, perfect. Can you also measure RNA contamination in a DNA sample? So the opposite. Yeah. So exactly the same. If you would working with DNA samples that have RNA contamination, it's a different application. It's called DNA tissue blood for mammalian samples mainly, but it will do pretty much the same indeed, finding RNA contamination within your DNA sample. All right, thank you very much. And how pure does my sample need to be? Well, that's one of the benefits on what we just showed, right? On a classic system, you would either need to measure a pure RNA sample to trust the total quantified amount, but you always take a look at those ratios and then maybe see they're off, but you don't know why. And so here we showed with different amounts of contamination that your samples actually don't need to be super pure to be able to measure them. So we can find DNA contamination and their protein contamination at different levels. So the benefit here versus classic UV-Vis is that you won't overestimate again anymore with a less pure sample. Great. Thank you for answering all those fantastic questions. And thank you again for the great presentation.
I also would like to thank you, all those who joined us live today. If you would like to have a deeper conversation with our team, please do get in touch with us at info@unchainedlabs.com or learn more at our website at www.unchainedlabs.com. We would love to connect with you. Thank you again for attending our virtual seminar, and I hope you have a great rest of your day.
Bye.
Bye.